in Arizona State University. He completed his postdoctoral fellowship training with Ali K at Harvard Medical School and Brigham and Women's Hospital. Before that, he received his bachelor's and master's in mechanical engineering and biomedical engineering from Tehran Polytechnic University. After that, he got his PhD in mechanical engineering from Virginia Tech. He has received numerous grants from federal and local agencies, including NSF, NIH, Flint Foundation, Arizona ABRC, etc. He has more than 70 journal papers, 8 book chapters, 90 peer-reviewed conference papers, and currently holds 5 US patents. He has 8,500 citations and an H-index of 43. He's received numerous prestigious awards and recognitions during his career, including Arizona Flint Foundation Award, ASU Biomedical Engineering Outstanding Assistant Professor Award, NSF Career Award, so forth. He has trained more than 60 mentees, including postdoctoral fellows, PhD, MS students, as well as undergrad researchers from diverse backgrounds. The floor is yours, Mehdi.
Thank you very much, Mehmet. It's great to be here, and then special thanks to you and Ali for this kind invitation. I just shared my slide. I don't know if you see my slides or not, but yeah, thank you for this kind invitation. And I want to just, do you hear me, Mehmet?
Yes.
Okay.
Slides
great.
Are good. You're good. Everything is
okay,
good.
Great. And I want to say basically congratulations to you and Ali for Ali and you and starting this interesting institute for biomedical innovations. It's doing great stuff, as I see and actually kind of like follow. Also, congratulations for this excellent seminar series, which is pretty significant when you kind of realize that this seminar series does not have any limitations or boundaries for specific people, specifically in the US, and it's everyone, students, faculties, either in academia or industry across the world can watch this seminar series. And actually, this is very good for learning and education of our specifically students and trainees. So great, and thank you so much. So today I want to talk to you about some of the work that we have been doing at Arizona State University. As Mehmet mentioned, I was trained in Ali's lab at Harvard Medical School from 2010 to 2014, and then I started my faculty position at ASU in January of 2014, and that was the time that we started this research. So the title is Microengineering of Organotypic Tumor on Chip Technologies to study cancer progression. So just a little bit of overview. This is the slide I show at the beginning of most of the talks that I give. The overview of what we do in our lab at ASU is we work at the interface of micro and nano scale technologies and biomaterials, such as hydrogel-based biomaterials, somehow a little bit synthetic polymers, and then we collaborate with a lot of clinicians, specifically right now in Phoenix area, and then biologists, fundamental scientists in biology, toward engineering of biomimetic and organotypic tissue models. And then our basically goal is toward 2 major avenues. One is disease modeling, and then one is regenerative medicine. And basically on the aspect of disease modeling, we have been very active in the past few years. And right now the lab is the pioneering state of Arizona on development of multiple tissue on chip technologies. We have developed breast tumor on a chip, brain tumor on a chip that I'm going to talk about, and then also heart on a chip, different cardiac diseases such as ischemia, heart attack, and LQTS2 on a chip, neurovascular unit on a chip in collaboration with our VA collaborators at VA Medical Center. And so we just, our studies is not just limited to development of technology platforms, although we have received numerous patents on just the innovations on the aspect of technology, but we kind of dive into the biology to find out what are the molecular, cellular, and tissue level mechanisms or signaling pathways that I'm going to talk to you about, which leads to the disease manifestation in the body, and how can we block these signaling pathways to kind of stop the spread of the disease. And as I mentioned, we have worked across numerous diseases, and one of the active areas that we have worked on is the tumor microenvironment. And this is specifically the reason for this is my background in cancer actually when I was a PhD at Virginia Tech. Then when I went to Ali's lab, I worked extensively on hydrogel-based biomaterials and microscale tissue engineering. Then I combined these two expertise toward the new avenue that I developed in my lab at ASU. So as what you see actually on this photo, on the schematic and cartoon-like photo on the left side, this is a breast tumor microenvironment, and then these are basically the ducts. These are the tubes which carry the milk to the nipple, and then one of the main causes of breast cancer in patients is ductal carcinoma in situ. That what you see, these epithelial cells or myoepithelial cells, they transition to kind of like disease cells. They form ductal carcinoma in situ or DCIS, and then they rupture the duct, and then they kind of invade through the surrounding tissue. What you see interestingly on this image is the presence of multiple cell types within the tumor microenvironment, such as these green cells or fibroblast cells. We'll get back to this. And then these purple cells are macrophages or immune cells. And then there's a very, very complex crosstalk between these cells, between cancer cells and fibroblast cells and the immune cells, not on the biochemical factors and growth factors or ligands that these cells secrete, which ultimately promote the cancer progression or metastasis or invasion into the surrounding stroma, but also on the biophysical aspect. For instance, the extracellular matrix deposited by fibroblast cells lead to the basically ECM remodeling and stiffness, which leads to the which promotes tumor invasion. So looking at this tumor microenvironment is a highly complex disease landscape, perhaps one of the most complex diseases that happens. That's why there's not a definite cure for a lot of solid tumors in the field, although there has been a great progress to reduce the burden right now, but still we have not cured this disease completely. So this because by nature, this disease is very complex. And there are multiple cell types in these landscapes. As you see, cancer cells, fibroblast cells, endothelial cells, immune cells. And then we have adaptive immunity, innate immunity, and then basically education of the immune environment to go toward pro-tumorigenic because of the signaling of cancer cells, which cause a lot of complexities in this thing. And then what is interesting at the beginning of this is the tissue architecture. So what, for instance, in terms of the breast tumor microenvironment, we have the breast duct, which has a nice circular architecture, and then it becomes basically dispersed and random. So the motivation of our research when we look at this disease landscape is that can we develop configurable and organotypic tissue 3D microengineered tumor models or tumor microenvironment models to mechanistically assess the influence of these stromal cells or immune cells on tumor progression. And the tools that we wanted to use to develop this organotypic tissue architecture, as I mentioned, is microscale technologies and biomaterials. So we looked at microfluidic technologies that I'll explain in a second, but this is a histology from a patient. This is the tumor, and this is the stroma. As I mentioned, these are epithelial cells. This is the stroma. So the question was, how with the tools that we have, how do we kind of recreate this nice architecture ex vivo or in vitro with patient-derived cells or primary cells, and then we can conduct our mechanistic studies. So this was, I'm going to actually go through the story as I go forward. So this was the early microfluidic chip model that we developed to mimic this architecture on the left side, on basically on this, as you see on the histology image. So if you cut this actually in half, you have a symmetric kind of like distribution of these cells. So we have designed the tumor. As you see, this is the microfluidic chip. At the core of this center of this chip, we have the tumor actually tagged in the red fluorescent dye, and then we have the stroma surrounding in a nice semicircular fashion, and then we have the media channel on the surrounding that we can infuse growth factors or drugs or whatever we want. And then, but interestingly, as you see on this middle image, a fluorescent image, the tumor and the stroma are interconnected while they're separated, meaning that at the initial injection of the tumor cells in the 3D matrix inside this basically region, you can actually have a nice circular architecture and then semicircular architecture. Then I can add the stroma later. So that actually creates the connectivity while they're basically crosstalk to each other, while they're kind of not mixed or randomly mixed, as opposed to kind of like most of the spheroid cultures in the tumor, or basically when you mix all the cells together, there's no architecture. So what we can designate a nice architecture. The other thing is like we can actually have any ECM matrix in the tumor or any cell type. You can mix immune cells with the tumor cells. and then have basically collagen matrigel and a stroma. We can add just collagen, we can mix it with hyaluronic acid. So you have all the flexibilities to play around this tumor microenvironment. So what you see on this phase contrast image on the below, so you have tumor cells here on the core and then stroma on the periphery. What you see, this is a nice architecture at the day 0. We can do all the COMSOL multi-physics simulations on the diffusion of nutrients and the growth factors and drugs into the tumor and establishment of that gradient to kind of like see what is the influence in the invasion. And as I mentioned, these are like, this has organotypic architecture. You can play around with the cells and the matrix and etc. So we have extensively published in this area from 2016 up to recently in 2022. And our technologies have been granted multiple patents actually. And then 2 active patents and the 3rd one is actually on the way. So basically this is the movie. As you see, this is the core of the tumor, that when we load cancer cells in a mixture of collagen and matrigel on the stroma, we have collagen. As you see these tiny kind of spots, these are cancer cells crawling inside the collagen. So this is not a 2D culture, it's a 3-dimensional culture that the cells kind of like invade through the 3D matrix toward the stroma. But now interesting is, if you look at these 2 movies on the, specifically on the right side movie, we can actually designate each one of these blocks is one single cell. So we can designate the block around the cell and measure or assess the migration of the cells in X, Y, Z, see how the cells kind of maneuver through X, Y, or the Z dimension of the 3D hydrogel. So this was a very interesting kind of like imaging that we did in the Z stack throughout the gel to see how the cells kind of move around. So our first study was to kind of spike the cells with epidermal growth factor, which is a growth factor released in the tumor microenvironment by mainly macrophages. So what we interestingly saw that this actually EGF significantly promoted the speed of the cancer cells toward the stroma, and also the persistence and directionality of the cells, meaning it was not significantly changed. So this was the first study. I'm just showing actually few slides on this that was published in scientific reports. We just wanted to show that we developed the technology. The technology is physiologically relevant, and we can do so many studies on that. Like, as I mentioned, we can measure single cell speed or persistence, and then we can create these tumor microenvironment and the technology actually, and it has the and showed its capabilities. But now, next, back to the tumor microenvironment. So that was a monoculture of the cells, just cancer cells. But now we want to make it one step further to make it more complex. So what do we do now? So one of the main axes of signaling which promotes the tumor invasion or cancer progression is between the fibroblast cells and tumor cells. But a lot of studies have looked at signaling of fibroblast cells in the context of tumor migration, but the problem is like either they have used lung fibroblast cells in the context of breast cancer, or they have used basically 3T3 mouse fibroblast cells, and then like or basically other types of fibroblast cell lines, not patient-derived cells. And also they have either mixed them in random co-cultures, and then the studies are not as detailed in terms of like what are the signaling pathways which promote the invasion. So with this, we actually collaborated with 2 fantastic colleagues, one Dr. Barbara Pachkai, who's an oncologist at Mayo Clinic of Arizona, and then Dr. Jim Park, who's a bioinformatics, who helped us like basically do all the bioinformatics work and gene expression with RNA sequencing toward the end. But anyway, so we actually isolated tissue samples from patients at Mayo Clinic which went under mastectomy, and then these patients were like basically either triple negative or ER positive, PR positive patients, and I'll discuss this in a second. So we would go to the pathology lab, collect the tissues from the patient, while the patient, at one case, the patient was in the surgery room while they were mastectomy, tissue came to the pathology lab, and then so we kind of like tell the pathologist where to take the biopsy from the tissue, actually, either from the core of the tumor or from 1 centimeter away from the tumor, 2 centimeters away from the tumor, or even the border of the tumor. So these biopsies were taken on the outside. This is the extracellular matrix, this cartoon image, like these are fibroblast cells. So we bring back the tissue to the lab, we digest the tissue and go through basically enzymatic digestion standard assays. And then we isolated fibroblast cells, but now we formed the co-culture tumor model on our microfluidic chip that we have cancer cells in the core of the tumor embedded in collagen and matrigel, and fibroblast cells are just collagen 2 milligrams per ml. But you see actually this table designates the patients. Actually, the patient 1 is 71 years old. He's She's ER positive, PR positive, and the patient 2 is 50 years old, ER positive, PR positive, and HER2 negative. And patient 3 was very interesting case. This is a triple negative cancer patient, which is actually one of the most basically aggressive types of breast cancer. Majority of the cases is very lethal, actually, type of cancer. So I just want you to kind of remember this patient 3, and then so when we isolated the fibroblasts. So after that, because of the limited availability of the patient and tissue samples, we were so lucky to get these 3 patients, especially the patient 3, because triple negative breast tissues are very, very actually valuable for different studies that the oncologists and breast cancer, basically biologists, they want to conduct these studies, but our collaborators were very kind enough to provide these tissues to us. So when we bring actually the tissues to the lab, so the first question that we want to ask is, can we just basically, whether these are the right cell types that we have isolated, specifically in this case, fibroblast cells. So we have here on this first panel, these are normal fibroblast cells that they have derived from bilateral mastectomy. So meaning that if one side has the cancer tumors, the other side has gone under mastectomy. So we isolate the normal fibroblast cells from the same patient on the other side. And then basically, so they're cytokeratin negative, vimentin positive, alpha SMA negative. So these are good markers to indicate whether these are normal fibroblast cells or activated fibroblast cells. This is cancer-associated fibroblast from patient 1, patient 2, and patient 3. What you see, these red markers here are alpha SMA. They're showing that there's an activation of the cancer cells. And the crosstalk between the tumor cells and the cancer cells, fibroblast cells, lead to the activation of the tumor cell, and there's a feedback from these activated fibroblast cells, which promote the tumor invasion. And then we actually have 2 types of breast cancer cells, MCF7, which are tumorigenic, and SUM159, which is highly aggressive cancer cell line, which is positive in cytokeratin and vimentin, actually. So now what we actually do, we can do the wound assay actually here to kind of look at the migration of these basically of the tumor cells, cancer-associated fibroblast cells, and then we can look at basically the wound closure percentage, and then we can do Western blot on alpha SMA. So all together, these studies are showing that basically the cells are kind of like the right cell types that we are kind of like isolated. Sorry, 1 second. Okay, so now we are sure that we identified the right cell type or isolated the right cell type from the tumor samples. We run our invasion assay in our microfluidic device. On the first row, just trying to kind of simplify everything. So it's just monoculture of tumor cells, and then day 1 and day 3, we see the invasion of tumor cells, this dashed kind of like a semicircular line. And normal fibroblast cells, you see invasion is inhibited, and patient 1, patient 2, patient 3, what you see actually, which is corresponding to this bar graph, as you see, these red cells are aggressive tumor cells, which are actually transduced with M-cherry, so we can track the migration of these cells. They're highly invading into the stroma, as also indicated by quantification of the migration distance, which was highest actually. And this was the patient 3 that I mentioned, that she was triple negative patient. So then when you get the fibroblast from this patient 3, put that into the invasion assay, it causes the significant amount of invasion of tumor cells into the stroma. But one thing which was very interesting that I want to actually notify is the normal fibroblast cells from bilateral mastectomy of the patient, they actually inhibit the migration. So this was a very opposing and interesting observation. So we have not had the kind of like opportunity to study why normal fibroblasts inhibit the invasion and cancer-associated fibroblasts significantly promote the invasion. So this was our first observation. And then now we did several rounds of cellular level studies, Ki-67, EDU, proliferation assay, cell viability assay, inside the microfluidic chip, migration assay, and then we looked at the movement of these cells. So, but this is just a summary of that. But then now, the question came forward is why this is happening? What are the signaling pathways? So interesting that our assay is compatible with a lot of biological kind of like assays or microfluidic assay, basically. So we can extract the cells using enzymatic digestions, and then we can sort just SUM, which are just cancer cells, from all the culture conditions that we had. And then these are M-cherry transduced, so they're red, and then we go through flow cytometry to sort these cells. And then we can do RNA sequencing on the cells and see what are the signaling pathways which promote the invasion of tumor cells. This is going a little bit deep into the biology now. And then, so what we actually see in the multidimensional scaling analysis, CAF co-culture groups are clustered together, and then SUM monoculture is clustered separately, and then the co-culture with normal fibroblasts. So this actually shows that the co-culture groups in the microfluidic device with cancer-associated fibroblasts clustered differentially from the normal monoculture condition or normal fibroblasts. But this is a heat map of the genes that we actually extracted from our RNA sequencing analysis. That shows actually this first line, or basically a row, is the normal culture, monoculture of SUM and normal co-culture with normal fibroblast cells. And then what you see actually, if you look at these basically green boxes, there's a differential expression of the genes when you have monoculture versus the co-culture with cancer-associated fibroblasts cells. So our colleagues, bioinformatics colleagues, actually helped us to go through multiple levels of statistical analysis to look at these differentially expressed genes. So at the first actually step at the hierarchical clustering, we identified 280 differentially expressed genes when we have monoculture of cancer cells versus co-culture with cancer-associated fibroblast cells. And now we actually went through those genes, and my student actually, Don Trunk, who's right now at the MD Anderson Cancer Center, actually he went through all these genes one at a time, and then through another level of statistics, we came to a panel of about 20 genes which were differentially expressed. So what you see, these are basically the name of the genes, as you see on the left side. So we have control versus normal fibroblast cells, control, which is the monoculture of cancer versus CAFs, and normal fibroblast cells versus CAF group. So we look at this differential expression of the genes, and then one of these genes out of these 20 kind of caught our eye, which is GPNMB. This is glycoprotein non-metastatic B gene, and actually encodes for a cell surface transmembrane protein. And this gene, there was a recent review article actually on this gene that it plays a lot of role in different types of cancers, such as melanoma. And then people have also noted that in breast cancer, but not in the co-culture of fibroblast cells. So our study was the first to report that this gene gets upregulated when you have patient-derived cancer-associated fibroblast co-culture with the tumor cells inside the microfluidic device. So when we identified this gene, the question comes, what is the physiological relevance of this finding that you have done in this ex vivo microfluidic model to the patient data, right? So now with the help of our bioinformatics collaborator, now we dig in back into the patient databases. So there are two databases. One of them is TCGA that everyone perhaps knows about, that we looked at a panel of 463 patients and 2,500 patients. But interestingly, we dig back into these databases, we see in TCGA 16% of breast cancer patients, this gene was highly upregulated, and in 10% of the basically cancer patients in the other database, actually this gene was amplified. And then now, what we actually look at the GPNMB mRNA expression in carcinoma tissue and normal tissue based on these databases. In the carcinoma tissue, which is simply cancer tissue, this gene is highly upregulated in these cancer patients. And interestingly, on this panel D, which is like basically overall survival, these patients that they have higher expression of these genes, they died faster actually. So this was a very interesting finding. And actually what we found in our microfluidic ex vivo tissue model or tumor model was well correlated to the clinical patient databases. And then, so basically, so after this, so we could wrap up the study and say, oh, okay, so we have found this gene, we have discovered that this gene gets upregulated because of the presence of the cancer-associated fibroblast, but then we decided to take one step forward. And then that was now, let's knock down this gene with shRNA. And then, as I mentioned, this goes a little bit into the biology now, away from the technology. But now we created 4 lines of shRNA knockdown of this specific gene in cancer cells. And then we did qPCR to make sure that this gene is successfully knocked down. And then this was work done by our excellent students. And then we look at basically immunofluorescence of this glycoprotein on the cell membrane. And actually we see significant downregulation of this basically protein expression of the GPNMB on the transmembrane due to the knockdown of this gene. And then that shows that our knockdown was successful. So now we hypothesize that we actually, if we add these knockdown cancer cells on this specific gene back into our tumor model on the microfluidic, we are going to blunt the invasion. So let's see what happens, which was very interesting. So we formed actually the monoculture of the cells And actually, so on SH control, so this is not knockdown line number 1 and number 4, we see a significant reduction in the invasion in the tumor ownership. Now, when we add cancer-associated fibroblast co-culture, even in the presence of cancer-associated fibroblast cells, when we knock down that gene, the migration distance of tumors significantly gets reduced. And now, on the last part of the study, we actually look at these two movies. On the left side, this is a control cancer cells that are invading through the tissue. And actually, when we knock down that GP and MB, they cannot actually invade throughout the 3D matrix. This is all inside the microfluidic device, 3D Z-stack movies generated at real time inside the device in the collagen gel. And then we can again measure migration speed. And you see the migration speed is significantly dropped. And this paper was published in Cancer Research in 2019 by our student Don Trong. As I mentioned, he's in MD Anderson Cancer Center now, but it was an excellent work which is completed. And actually, we discovered that this gene is upregulated because of cancer-associated fibroblast cells, which the knockdown actually can blunt the invasion, so it can stop the invasion. So that actually leads to kind of like could lead to potential to new avenues that whether we can actually design drugs that we can actually knock down these genes and actually to blunt the invasion in clinical data, in clinical basically samples. So, so the next part of the study, the question was that if you look at this axis of the fibroblast and tumor cell basically signaling, how do you study the biophysical influence? Here you looked at the signaling, basically chemokine or cytokine signaling between the two cell types, but what is the role of biophysical changes in the matrix or the tumor microenvironment? So we said the answer was yes. Now we can develop another tumor model, not a microfluidic tumor model, because microfluidic is enclosed, so you cannot actually probe the matrix. But now we can go a microvell-based tumor microenvironment model. We can have CAFs or cancer-associated fibroblasts in the collagen matrix, and we can form the tumors on the basically these circular kind of like microvells. And this has been used extensively in other fields of tissue engineering, such as embryo body formation and etc. But we use it now for the tumor. But now our colleagues in physics department at ASU, they are experts in AFM or atomic force microscopy. So they can come with AFM and probe the matrix because it's an open top and see what is the change in the stiffness of the matrix. So what you see actually on these, on day 0, just MDA, MB231, the cells are kind of nicely clustered inside these microvels, showing the formation of the tumor. When you have monoculture, they kind of disperse at day 4, invade into the tissue. But when you add CAFs, cancer-associated fibroblasts that I was just talking about, these are the cells which are not non-transduced. So this is the overlay image of the phase contrast fluorescent imaging contrast, and you see because of CAFs, they kind of like significant. So this was in accordance of what we found in our microfluidic device. And then these are Delanoid triangulation actually technique, but which was developed by another excellent PhD student, Harpinder, who's at the Bristol Myers Squibb oncology company right now. And then so then we can actually come and look at ECM deposition, extracellular matrix. We have MDA, MCF7 at day 0, day 4 monoculture. But I just want to draw your attention to these basically red dashed boxes. Actually, when we co-culture these cancer cells with cancer-associated fibroblasts, we see significant deposition of ECM or extracellular matrix as also defined by this. So that shows actually not this invasion or significant invasion of tumor cells is not just necessarily due to the cytokine signaling, but also there's a desmoplasia or basically kind of like excessive disposition of the extracellular matrix into the tumor microenvironment because of the cancer-associated fibroblast cells. Now, as I mentioned, our collaborators in physics came with their AFM, atomic force microscopy. So we form the tumor, we give it to them, and then now they can come and probe actually at different days and see what is the matrix change, stiffness changes. So these are just different groups. I just want to simplify this. So these are monoculture of blue red bars showing the monoculture of tumor cells, and then we have MCF7 tumorigenic cells, and then we have CAFs, cancer-associated fibroblasts. What you see at day 0 to 4, there is no significant change in the stiffness, even with the presence of just CAFs that are actually responsible for deposition of ECM. Now, if you just have CAFs in the matrix, nothing is going to happen in terms of like stiffness. But as soon as you co-culture tumor cells with CAFs or cancer-associated fibroblasts, we see that significant increase in the stiffness of basically of the matrix. So let me see if I can put the laser pointer actually. So that's better actually. So we can see actually the significant increase in the migration of basically, stiffness of the tumor matrix because of just co-culture of these two cell types. So that means that when we have monoculture, nothing happens in terms of the stiffness matrix, stiffness of the matrix. But as soon as we actually co-culture these two cell types in our assay, there's a significant increase. So this was in accordance of our previous studies in the microfluidic device to show that there's basically significant invasion of the tumor cells because of fibroblasts. So this was the fibroblast story that we developed and validated at the biochemical, biophysical kind of like aspects, also validated with the patient databases and etc. Now, with respect back to our microfluidic, so on the previous basically study, we have tumor cells and we have stroma where we added tumor cells and the CAFs, cancer-associated fibroblast cells, into the stroma. Now, the question came and people started asking that tumor has this angiogenesis and formation of blood vascular networks. And I personally worked on vascularization specifically when I was in Ali's lab. So we kind of like said, okay, why don't we kind of enhance this tumor model and make it more complex by adding a 3-layer inside the microfluidic chip. So we have tumor, we have stroma, we have a vascular region. Now, again, this architecture is organotypic, and then which has the tumor on the core, stroma on the periphery, and the vascular on the outer level. You can add any cell types inside this region. You can add any matrix you want. So, for instance, our vascular region, and I'm going to show actually on the next slide what is the matrix, and also is in fibrin, and then stroma could be collagen, tumor cells, basically region could be matrigel, and you can add any cell types. You can add immune cells into the tumor region and stroma that we are doing right now. And now, what you see actually this image on the left side, what we see, these are capillary networks that are formed by these endothelial cells inside the microfluidic chip, and then these red dots are the cancer cells. So simply cancer cells couldn't go inside the fibrin gel versus they intravasculated or went inside the blood capillaries. And then this actually movie is showing, this movie is showing the cancer cells are kind of nicely kind of crawling inside these. Let me see, how can I do this? Okay. Oh, so sorry. I think I clicked something here. Okay, so I'm back to, sorry, issues here. So anyway, so we actually, performed this, and then this was specifically on breast cancer, and then our studies, we were granted another patent on this technology that we are further advancing that, as I mentioned, to induce immune cells on this microenvironment. But now on the last part of the talk, our excellent collaborator in Barrow Neurological Institute, which is the brain, and they have a brain tumor center in Phoenix, and it's one of the active brain tumor centers in the nation. And they approached us and I said, okay, we have observed this publication coming out on mainly on breast cancer. Why don't we develop a brain tumor on a chip using your technologies that you have developed at ASU? So we kind of, after like several discussions, we came up with the development of a brain tumor on a chip. As you see on this cartoon image, within the brain tumor microenvironment, there are different niches, that all these niches promote glioma stem cells invasion or brain cancer, brain tumor cell invasion. So these niches are either hypoxic niche, so we have formation of these clusters of basically hypoxia or hypoxic regions in the brain tumor microenvironment, or it could be an invasive niche, or another niche which is really important, the perivascular niche, and then we have blood vessels actually, and then all these brain tumor cells are actually basically attracted toward these blood vessels. So what we actually said, so can we create this perivascular niche on the chip? And then the answer was yes. So we came back to the same microfluidic device that we have. So 3 layers. So we have the tumor on a chip on the center, like basically tumor cells at the core. And then we have stroma on the periphery, and then we can have the vascular region. This is the chip. Actually, is that we have is next to the dime, and then we can actually go back and do our studies. So this is basically the vascular region, which is formed on the outer layer. So these are lumen formation. And this movie, if you pay attention to this movie, we did 3D confocal imaging of basically of the 3D confocal imaging of basically of this vascular network inside the chip and showing the nice formation of the lumen. Next, we want to look at some movies actually of these brain and brain tumor cells. That these are the microposts in the order of 200 micron inside the microfluidic chip. This is the green area, which is the vascular region, and we do confocal imaging and see nice and extension of these brain tumor cells toward the perivascular niche. And actually, these are, as I mentioned, these are the lumens of the basically endothelial cells that we are going inside the vascular region. And I consider these images are in the order of basically 30 to 50 micron that we have taken images, or at the order of a single cell inside the chip, that we can basically precisely see what is the cell-cell interaction between the tumor cells and the vascular niche using this technology. So which was pretty interesting. So these are the cellular observation. And then we have done several studies that I'm not going to show here in terms of migration analysis, proliferation, stemness, that I'm going to show next on the last part of the slides, but also what are the medial composition that we need to add in the triculture or co-culture to kind of like have basically an optimized assay for our invasion analysis. But on the last part of this study, on the that we have PDX, patient-derived xenografts, and then the same GB3 cells, which are glioma stem cells, a subset of glioma stem cells, and these are patient-derived tumor cells from the brain. They're orthotopically actually injected into the mouse. And then what we see actually, these red cells, which are GB3 cells transduced with RFP, red fluorescent protein, and actually they're kind of going nicely along these blood vessels, which are actually tagged or stained in lectin. And then now this is the top row. On the bottom row, we have our microfluidic device. We kind of see the same observation. So this was another checkpoint of the physiological relevance of our microfluidic chip to see and confirm that whatever we observe in our microfluidic chip is in concordance of whatever we see in vivo within the animal model. So the next, this was published in Biomaterial on co-culture of endothelial cells and brain tumor cells. On the last part, we actually said, so why don't we make this brain tumor even more complex? So we started the simple model and then now add the complexities of this model system. Now we added astrocytes now. So we have monoculture of the tumor cells. We have astrocytes on the stroma, endothelial cells, endothelial cells. And now we have astrocytes in the stroma, tumor cells at the core, and endothelial cells. So we can go from monoculture to co-culture to the triculture. And then now astrocytes is one of the prominent cell types in the brain tumor microenvironment. So this is the key kind of like presence in the tumor invasion. So we form this triculture. We can do so many things. We put it under 2% oxygen in the monoculture, and triculture, we can see that the cells are live and then doing very well to kind of like mimic that hypoxic niche. In addition to the perivascular niche, and then we can look at the invasion of these cells. And actually what we noted that when we have brain tumor cells, there's a synergistic influence of endothelial cells and then astrocytes, which promote the invasion of the tumor cells, or brain tumor cells into the stroma. But then the last part of the study, I want to take perhaps 5 more minutes and I'll stop, is that what we did actually to kind of confirm what are the molecular pathways which promote the invasion of these tumor cells. So first, we kind of like stained the cells for basically for neural progenitor markers, which is the nestin, and then these are RFP, red fluorescently tagged basically cells, tumor cells, and then they're expressing nestin. This is the co-localization of the nestin. And then we look at SOX2, which is the stemness marker of these cells. So this is the monoculture on the first row. On the second row, we have co-culture with astrocytes. Then we have co-culture with vasculature, and we have triculture conditions, which has the endothelial cells and the astrocytes. And CD44 is actually HA receptor and GBM, glioblastoma marker. So what we see actually, as we noted in the triculture, we have nice extension of these cells. So they're extending their processes as we showed in the movie. But the overall message of this basically slide is that these glioma stem cell patient-derived brain tumor cells, they actually maintain their stemness phenotype. And then they kind of migrate toward the vasculature based on these images. Now, as I mentioned, this was cellular level analysis. Then we advanced it further with our colleague in bioinformatics and said, okay, why don't we now take the cells out of the chip after we make our observation and then see what are the molecular pathways which promote the invasion of these brain tumor cells into the stroma because of the perivascular niche or because of the presence of the astrocytes and etc. So these came to single cell RNA sequencing, and then so we extract, similar to the studies that we did for a breast tumor on a chip. Now we kind of extract the cells from the chip, and then now instead of bulk RNA sequencing, and then which doesn't lead to identification of specific pathway, we can do single cell RNA sequencing. So what you see on the top, we have monoculture of basically glioma stem cells inside the basically tumor region, and we have 3 monoculture of the cells like glioma stem cells, astrocytes, and endothelial mixed together at day 3. But now, we let the cells kind of interact with each other inside the chip, and now we digest the tissue and take the cells out, and then we see formation of nice clusters of the cell, showing that we have identified the right cell type in our single cell RNA-seq analysis based on markers such as endothelial marker PCAM1 and then S100D for astrocyte marker, as CDKNA2 for GSC markers. So this was just showing that the cells are basically based on unsupervised clustering. We can identify these 3 cell types in our triculture. But now, last part, which was very interesting, and then it was okay. So these cells are interacting inside the chip, and we have seen an enhanced invasion or migration of these tumor cells because of the presence of endothelial cells or astrocyte or synergistic influence of these cells. What are the ligand or receptor kind of pairs which are secreted in the tumor microenvironment, which indeed promote the tumor invasion? So in other words, what are the ligands secreted by either endothelial cells or astrocytes or even GSC in a self-feedback mechanism which promote the invasion? So what you see in this cartoon image, so we have HUVECs that they secrete these ligands in form of triangle, and they diffuse through the stroma, diffuse through the tumor. These are human glioma stem cells. These are patient-derived brain tumor cells, and they have receptors to different ligands that are either secreted by endothelial cells or astrocytes in the stroma region. So this is a vascular region, stroma region, and the tumor region, and these ligands are basically going from normal human astrocytes, diffuse through the basically stroma, and then go into the tumor region. So we can kind of mimic very nicely what's happening inside the body using this microfluidic chip and the technology. But now, after single cell RNA sequencing and extraction, our bioinformatics collaborator and the excellent PhD students kind of like paired with our PhDs to identify what are these ligands. So what you see on this image, which is kind of like a little bit complicated, these receptors, these are showing the receptors, and actually the monoculture condition are basically in orange, and then triculture are actually tagged in purple. And then we have HUVEC, we have astrocyte, and we have GSC. So after multiple levels of basically statistical analysis, first we identified a set of 33 receptors on GSCs, then we narrowed down to 17, then we narrowed down to 6 prominent receptors which promote the invasion. So we have all these receptors which are actually expressed by GSCs, which are brain tumor cells. Now we can look at the ligands. What are the ligands which correspond to these receptors and where they are coming from, either from coming endothelial cells or astrocytes? So these are, for instance, for platelet-driven growth factor A, which is kind of like the receptors, we have PDGF-A, PDGF-C, PDGF-B, and these are all the ligands. And then we can show where these ligands are coming from. For instance, they're coming from here, the red boxes are showing that they're coming from astrocytes. But now we looked at these, and then the interesting part was that some of these ligand receptors has been previously established in the literature. So that shows the kind of like validity of our model, that we can not just discover a new pathway, but we can also first identify and confirm a pre-existing pathway which has been shown in the past, but now we can go toward the discovery approach. So now we digged into the clinical databases of the patients, like these are different databases of patients that they have brain tumor. And then now we have the grade 2, grade 3, grade 4 tumors. And then interestingly, when we look at these boxes, dashed boxes, there are 2 ligand receptor pairs that we identified, which was not investigated in the past. So one is FPR1, SAA1, which is the ligand, and the other one LGR6 receptor, and then RSPO3 ligand. And both of these basically ligands and receptors pairs are actually expressed in the patient tumor basically databases, similar to what we actually observe in our breast tumor model. So basically what we actually said, okay, so in the triculture condition, we have identified these ligand receptor pairs. We have validated that based on the clinical databases of the brain tumor in patients. Now let's go back to the microfluidic model and then validate that further. So we went back into the microfluidic model, and then now we have the control condition in monoculture. So we remove all the stroma and vascular cells, and we just infuse the ligand through the media channel of this microfluidic cell, microfluidic chip, to see what happens to these brain tumor cells. So we have the control on day 0 and day 3. So the cells kind of invade, and these are the processes as you see. When we add this ligand RSPO3, we don't see a significant invasion, but when we add SAA1 ligand, we see a significant invasion of the tumor cell in monoculture conditions. So meaning that there's no co-culture or triculture. We identified this ligand from triculture condition, and then we took that exogenous ligand back to the monoculture into the chip, and we showed that there's a significant invasion because of this ligand. So in our next step, this was published recently in Advanced Science, if you want to look at the literature, but our next step is like, now what if we kind of design drugs to block this pathway in vivo, and then we can kind of like see whether we can blunt the invasion of these tumor cells inside the brain tumor microenvironment. So with this, I think I would like to stop here. This was the last slide showing our stories on the breast tumor microenvironment development. So we developed 2-3 sets of the tumor model for different needs. So one of them is basically to assess what are the signaling pathways at the cytokine level between the co-culture of stromal cells and tumor cells or immune cells. And then we identified a novel, basically genes which promote the tumor invasion. And then now we can advance our microfluidic chip to a tri-layer model by adding a vascular region. Now we can apply it to different types of tumor. Here, we applied it to brain tumor. And then that led to interesting discoveries that I just talked about. But now our studies have not been just limited to tumor microenvironment models, because one of the aspects of tumor microenvironment is the fibrosis, that we were very interested to look at what fibroblasts do to the tumor cells invasion. But there are similarities of the content in terms of the biology context to other diseases. So for instance, we have, as I mentioned, we have developed heart on a chip model, and then we have mimicked heart attack on a chip that I don't want to talk about this here because it's out of the scope of this talk. And then we have shown the activation of fibroblast cells, and then because of presence of fibroblast cells, the manifestation of the disease, and then and etc. So we have developed multiple kind of diseases in the avenue of cardiovascular that kind of somehow related, but in terms of like technology development or biological discoveries to this fibroblasts. So anyway, this work was not possible with excellent students that we have had. This is the past students that we have had on and helping there on basically the tumor microenvironment model. And now the new students that we have had, some of them are working to add basically immune cells and make even the tumor model more complex. And then we are trying to see how can we kind of adjust these tumor models to kind of for high throughput drug screening and kind of like push it toward commercialization and avenues. And I want to basically thank you, the funding agencies from NSF to NIH, for supporting our work. So I'll stop here for any question. Thank you so much.
Thank you for the great talk, Professor Mehdi. I appreciate it. Tumor cells are actively growing, and space is fundamental growth limiting factor in artificial tumor culture in Petri dish or your model. How do you want to deal with this problem? Do we need to follow specific cell number or timing in order to have correct result and minimize space effect on the experiment?
This is a very good question. So what I showed right now on our breast tumor on a chip, I just showed the results. But the first assay that was successful and worked, it took us about 7 to 8 months to kind of optimize that assay. So what are the optimization factors? Concentration of the hydrogel that we inject inside our chip, apart from the chip design itself. What is the cell density? And that's a very valid question. So our cell density is right now is 15 million cells per micro ml of the hydrogel, but we just inject 3 microliter of that. So meaning that in each chip, we end up having about 50 to 100,000 cells. So it's a very low number compared to kind of like larger assays that people use. But there was an optimization that our students did, because if you add too many cells inside the tumor, they're going to overgrow and they're going to die. And then you may see formation of hypoxic core at the center of the tumor. If you add low number of the cells, then that limits the invasion analysis. So there are like steps of optimization in terms of cell culture, or another step of optimization is the media. Like when we have co-culture or tri-culture, we cannot just add simply the media that we use for cancer cells to endothelial cells. For our tri-culture model on the vascular region, when we have the brain tumor model, for instance, we grow endothelial cells for 3 days, and then in the endothelial specific growth media, which is EGMT bullet kit, and then we take out that media from the chip, and then when we add cancer cells, we have to add a cocktail of combination. So these are different steps of optimization that we have to go over. But that's a good question.
Thank you. Have you tried to use paired patient-derived CAF and cancer cells in a device?
In terms of patient cells on the breast tumor, we isolated fibroblast cells from the patient tumor, as I showed, and then we actually we are in terms of immune cells, we are going to isolate monocytes from blood, basically samples of patients. And then in terms of glioma or stem cells or brain tumor, we actually just isolated these glioma stem cells, which are brain tumor cells. They're derived from different type of patients. And then we can run our studies. So this was basically it. But yeah, I hope I answered that question.
Yes, yes. Since cancer, thank you for this insightful presentation. Since cancer-associated fibroblasts sometimes suppresses T cells, why you didn't add immune cells to your study for breast cancer on a chip? Is it challenging to do?
That's a very good question, actually. So going back to that optimization study, just adding one cell type. So when you have monoculture, the assay is very simple. That was the first paper and first part of the story that I showed. When you add the second cell type, the complexity exponentially increases, like in terms of media, cell number, density, what matrix do you want to use? Right now, after we did all this work, our most complex culture is on the brain tumor that we have 3 cell types, and now we are moving to quad culture right now by adding another cell type. But in the breast tumor microenvironment, one of the active projects that one of our students is working is adding macrophages first. Then we want to add T cells, actually, cytotoxic T cells. And then, so definitely there is an opportunity, but we wanted to complete this first part of the study. And frankly speaking, we just don't want to like add cells and then do some cell level analysis and then just send the basically study out. As I showed in these papers, in these manuscripts, some of them took a couple of years or 2 years, especially on the paper on cancer research. Our intention is to just dive into the biology and then make discoveries. So that takes a lot of time, apart from optimization of the technology itself. So, but that's the avenue that we are actually pursuing right now by adding immune component to our tumor microenvironment.
Thank you. Thank you for a nice talk. The tumor vascular model has the vascular region on the outside, whereas in vivo, blood vessels run through the tumors and not on the outside. Can you comment on this?
We can, yes. There definitely, there's a limitation of in vitro assays. So we have seen spheroid models that they kind of like mix endothelial cells with tumor cells and then just put that as a spheroid. And then we can see kind of like basically sprouting of this network. But for us, what was important was, of course, there are some angiogenesis toward the tumor, mass itself. But what you see, as I mentioned in that image of the dog, from the breast tumor microenvironment, initially the tumor are at the core, and then you have the stroma, and then angiogenesis at that initial stage may not have been all promoted to kind of like penetrate into the tumor. So that's why I call this an organotypic architecture at the initial stage, although after that the cells are going to be intermingled. So our goal was like to have an organotypic architecture first, and then we didn't want to just mix all the cells together into one region and then have all these kind of random co-culture or mixture of the cells into one region, which does not let us to do our well-controlled studies. Like we wanted to have this nice organotypic architecture and then basically monitor at every stage of the tumor progression. So that was the reason for our design. But definitely there's an opportunity to do that, but we have to keep in mind that in vitro assays have always limitations. I'm not saying this is the best, like excellent 100%. So for instance, if you want to mix endothelial cells with tumor cells inside the matrigel, or collagen, endothelial cells, they won't form sprouting. So you need to add fibrin to that. So these are the limitations that you have to work around to develop the best ex vivo assay for your studies, for your target studies.
Thank you. For brain tumor studies, you use CD44, a marker of stem cells. So you were detecting cancer. Are you detecting cancer stem cells? So fibroblasts are promoting stemness?
So for brain tumor microenvironment, we didn't use fibroblast cell to just clarify. We use fibroblast cell in the context of breast tumor microenvironment. For our brain tumor, what I wanted to show by CD44, these tumor cells are derived from patients. These are called glioma stem cells. So what happens in terms of glioma stem cells? They have a stemness phenotype, okay? And now what happens within the brain tumor microenvironment, when they go to the proximity of the vascular niche, as we also showed in the microfluidic device and in PDX, we wanted to see what happens to these cells, whether these basically cells with stemness phenotype, namely GSCs, they kind of differentiate to a specific subtype. And we were interested to see that in the presence of astrocytes and endothelial cells, or no, they maintain their stemness. So in that specific study of the triculture, we found that they remain, they keep their stemness phenotype, meaning that they're like they have the markers for neural progenitors and then stem cells. So that was the reason for that. We actually also stained for astrocytes markers to see whether these GSCs or glioma stem cells specifically differentiate to astrocytes or not, and we didn't observe that. But maybe when we add another cell type, such as, for instance, immune cells or like, etc., fibroblasts, to some extent, there are very little in the brain tumor, but that may happen too. But that was the whole goal of this, basically staining on that slide.
Thank you. One final question. Can you share if your tumor vascular model experiments have been performed in microgravity environment? Are there potential benefits to therapies in microgravity environment, such as affecting tumor cell migration patterns?
That's definitely an area which is kind of a lot of colleagues right now are pursuing. And as you see, there are different initiatives by NASA to see how the microphysiological systems kind of behave in the gravity, in the low gravity condition, and what can we learn from that, bring it back here in terms of like studies. For us, we have not done that for this specific study and the chip. Everything has been done here in the lab, not in the microgravity condition, but definitely the chip has the capabilities to do that. But we can, as I mentioned, these chips are capable of, for instance, putting under hypoxic environment. So as I showed, in our work of our cardiac, actually, heart on a chip model, that publications came out, and there's a new publication is going to come out soon, which got accepted this last week. We have actually put the chip under different hypoxic or oxygen gradients. And then now we have added CRISPR-Cas9 gene-edited cells to create a specific disease phenotype on the chip. So there are like multiple kind of like flexibilities and avenues that we can actually pursue, but specifically we have not done that in the low gravity condition, but there are opportunities if you do that, as shown also by other colleagues. If you do that, you can definitely learn several interesting factors that you can actually be useful to study the biology of the disease.
Excellent. Well, thank you so much for a fantastic talk, and so many questions came in. I appreciate your time, Professor.
Thank you so much, Mohammed, to you and Ali again, and all the institute for this great opportunity. I hope you're having a great day, and thank you for the great question. Thank you so much.
Okay, have a nice day.
Have a